In taking action to address one of the key strategies of the Brain Cancer Centre around critical mass, we've recruited three incredible, and don't take my word for it, I'm sure you'll know after this as well, three incredible young people who are all focused on brain cancer, who come from very, very diverse backgrounds. This tripartite leadership model is something that we know has worked in other cancers and where disciplines come together to create new ideas and strategies. It's my pleasure to welcome Dr Saskia, Dr Sarah and Dr Jim, who are here to talk about the Brain Cancer Research Laboratory. So welcome.
Thanks, Sam. And I hadn't seen any of that video before, but I have to say, stepping up now, I'm feeling really quite energised after seeing some of those pieces. So it was great to see it fresh today, just before we get a chance to address everyone here and talk about our vision for the future. So it's a great pleasure for us to be here today and to have an opportunity to describe how we are working together to address the challenges of brain cancer. So first of all, we'll each be taking you through our background, our diverse skill set, and how these complement each other to provide a vision that combines fundamental biology, applied bioinformatics, and clinical translation with a focus on improving patient outcomes. And this unique approach allows us to build a program that will take the learnings from other cancers and develop new tools for analysis and preclinical platforms, to develop the use of bioinformatics and machine learning for new stratification of samples from patients, and importantly, within our team, but also part of the broader mission of the Brain Cancer Centre, to identify new treatments and combinations for patients. Now, you've heard the mission described this afternoon, and I think reiterating it here, we want to see a world where brain cancer is a treatable disease and survivable disease, where no patient's lives are lost to brain cancer. And to deliver in this vision, we acknowledge the tension here between the urgency of hope and opportunity now for patients with a lived experience, as well as an understanding that there will be a need for long-term research and focus to deliver on this vision. And for this to be successful, new approaches and a new team approach is required. And that's where the model of having, the three lab heads coming together, working with one vision, can hopefully accelerate this process. But we're only just a small part of the team, but we're really excited to work more broadly. My background as a clinician scientist is to really sit at the intersection of the discovery work and the clinic. And I've got a particular focus on novel clinical trial design on the brain perioperative platform, which I'll provide a little bit more detail about later. Something that together with Professor Mark Rosenthal and Professor Kate Drummond, we've been trying to achieve for years. And it's really taken the establishment of the Brain Cancer Centre and the vision of the Victorian State Government for this to happen. And as well as thinking about survival, and that's a key goal for us, it's really important that we focus on the key determinants of quality of life, and that's the centre of the work that we do.
Thanks, Jim. I'm Dr Sarah Best, and I'm a cancer research scientist. My research training has focused on the biology of tumour cells, what they use to help them to grow, and how we can target that fact to develop a more personalised approach to treat cancer. I bring my knowledge and experience from different types of solid tumours, from breast cancer to skin cancer and lung cancer, and now I'm applying these skills to brain cancer research.
All right, hi, I'm Dr Saskia Freytag and I'm a bioinformatician. So a bioinformatician works at the intersection of statistics, biology, and computer science. And we are there in order to analyze the tremendous amounts of data modern experiments produce. So during my career, I have used statistics, machine learning, and high-throughput sequencing data to make really interesting discoveries in neurological disorders such as epilepsy and brain malformations. I've also applied computational methods to deepen our knowledge of how the brain develops from when we're in the womb all the way to when we are old. And now I'm using my knowledge of bioinformatics and brain development in order to solve brain cancer so that one day no lives will be lost to brain cancer. All right, so now that you've heard about us, let's introduce you to our team. So we have 3 wonderful individuals that make up our team, and they're really talented and dynamic. So we have Zach, who's our unicorn postdoc, and he works in both the lab and in front of the computer. We have Shannon, who manages our lab with her absolute wealth of experience. And then finally, we have Joel, who's a really talented and excellent research technician who's also working in the lab and in front of the computer. And like Sarah, Jim, and I, they bring really specialized knowledge from their fields that they have been doing previous research in, and that really have shaped the direction our own research is taking. So as a team, we hope to approach our research with a modern lens. And that means that not only the 3 of us work together every day, but our team works collaboratively. Given our different backgrounds, this means that we have to learn from each other and have to go outside of our disciplines. And that means we have to embrace diversity to deliver a truly multidisciplinary approach to research. Beyond this, we are really conscious that the lived experience of brain cancer can only enhance our research. And so we have one of the largest patient advocate teams at WeHide that consists of brain cancer patients as well as their carers supporting us. To really reflect these values, we very recently had a day where we brought our collaborators together, our team, as well as Paul Malouf, who's in the audience today, who is someone with brain cancer. And we were able to learn from his journey and then co-design our research vision together. We're always on the lookout for individuals to join our team. So I wanted to tell you about how the lab values that we have are reflected in the way that we train the next generation of scientists. So we want everyone in the lab to wear 3 hats. We want them to be able to work in the wet lab, so in the laboratory. We want them to be working in front of the computer, or what we call the dry lab. And we want them to be able to translate any discoveries that they're making to the clinic. And to facilitate this, we have the help of WeHi's excellent education program that oversees the training of all students at WeHi. Additionally, through the brain cancer centers, our students will have access to additional opportunities that will really ensure that they are the individuals that are making discoveries for brain cancer in the future. So what do students, the team, Sarah, Jim, and I actually focus on in our research? I'm sure that's what you're here to learn about. So our research vision consists of 3 programs. Our first program aims to identify better treatments by acknowledging that brain cancer comes in really a lot of different types and shapes and responds to a lot of different drugs. And by grouping brain cancers according to their molecular properties, what we're hoping to develop is the right drug for the right patient. In the second program, we're going to try to learn about the brain and the immune cell types that reside at the tumor border, and how they interact with each other and how they interact with the cancer. And we hope that this improved model will allow us to treat brain cancer better in the future. And in the third program, we're trying to build better preclinical models of brain cancer that really exemplify the diversity that brain cancers come in, and that will then allow us to test treatments and make sure that they're effective in the future. I hope that you can appreciate that these core programs are really conceived with all our expertises in mind, and that means that when we try to achieve them, what we're going to have to do is we're going to have to work together, learn from each other, and grow together. So our core programs are not only informed by our expertise, but also the technological advancements the scientific community has witnessed. To give you an appreciation of how far science has come, I'm showing you the really early days of microscopy, where the scientists had to rely on their ability to sketch and resolution was really poor. Today's microscopes do not just allow greater magnification, but they can reveal molecular properties of different cell types. So here, for brain tissue, These technologies can deliver these incredibly detailed images that can show you how molecules are expressed even at the subcellular level. So that means in different compartments of a single cell. And these technologies will allow us to understand the complexity of brain cancer at a completely new level and ensure that we develop effective drugs.
And so, in order to use these exciting new technologies, we actually need to build a lab. And so I want to take you through some of the milestones that we've achieved since the lab was established in October last year. First, as Saskia has pointed out, we recruited some wonderful and talented team members, and come Q3 this year, we'll also onboard a PhD and a master's student. Excitingly, for me at least, was the arrival of our first set of pipettes, and all the scientists in the room will appreciate that. And these help us to work from the most basic of experiments through to the most serious technology. They're absolutely essential. We spent a lot of time getting administrative goals off the ground and have been working on protocol development and building our first projects. And now we have a bustling laboratory. So in order to build and develop more exciting projects and hire more wonderful people in the lab, Jim, Saskia and I have been busy writing grants, which is how we get money to do more and more projects. But one of the early grants that we received was to use one of these exciting new technologies that Saskia was mentioning, and it's called Nanostring. So I couldn't have you come here today and not show you some actual real-world science data. This is what I do. So in this project that I'll show you now, Jim, Saskia and I were curious to investigate the differences in tumours that look homogeneous, or they look the same. This is an anaplastic astrocytoma brain cancer that was removed from a patient at the Royal Melbourne Hospital who consented for their surgical resection to be used for research. With the help of Jim and his clinical partners, we identified this sample for the project. As you can see, when we zoom in on a region of the tumour, all the cells look the same, which is even clearer when we look at the expression of a tumour marker called GFAP. Next, we investigated the expression of Ki67, which is a marker of cell division. With the new technologies at hand, we wanted to investigate in this tumour, where all the cells look the same, what are the properties of the cells that are rapidly dividing in brown, compared to those that are not in blue. And so we could separate those cell types using the Nanostring platform. So by looking at the expression of our key markers in the tumour, GFAP for our tumour cells, CD45, which marks our immune cells, and Ki67, which is the marker of cell division, we could isolate individual cell types in the tumour, so our non-dividing tumour cells, our dividing tumour cells, and our immune cells. And from these individual cell types, we could investigate the transcriptome, which tells us all the genes that are active in these different types of cells. Through this analysis, we identified a region of the tumour where there was particular enrichment of dividing cells and those that are not. So this highly dividing area in the top of the Ki67 graph. So we could analyse these regions individually, and through the bioinformatic expertise of Saskia, we identified that in the less aggressive region of the tumour at the bottom here, there are more immune cells that are capable of patrolling and killing tumour cells, which are these cells in this red and blue bar. We also found that in the aggressive region of the tumour, there was an increase in blood vessels, which is consistent with the idea that these aggressive tumours need more nutrients for them to divide rapidly. So next, Saskia investigated the properties of the tumour cells that were rapidly dividing. And the properties of these cells will help us to identify new drugs that will target these aggressive tumours. And we found that the gene expression in the aggressive part of the tumour was very different to the low dividing region, with interesting gene networks involved in neurogenesis and regeneration activated. So we've recently been approved by Nanostring to extend on this project, and we're really excited to see what new pathways we identify to target these aggressive tumours.
I'd also want to sort of take an opportunity to describe how the discovery programs will hopefully rapidly translate into the clinic. And that it's really important that the discovery programs link very closely to data collection and clinical trials, and then that whole feedback loop, as Sam and with Doug have described earlier, continues to iterate and learn over time. The current challenge is clear, and that is that the pipeline of phase 1 to phase 3 clinical trials has really failed our patients over the last 20 years. And fundamentally, this is because of a lack of understanding of what happens in the laboratory system and translating that to patients. And that disconnect has meant that we haven't made the discoveries where other cancers have had successes. So the opportunity for perioperative trials, that is where we collect patients' tumours at diagnosis and then enrol them in very unique clinical trials and treat with a panel of drugs or radiation or another therapeutic, and then collect further tumour samples on treatment, provides a really unique opportunity and asks two very important questions. Do drugs get to where they need to go? And what is the effect of drugs in the brain? And with the support from the Victorian government and with some leadership from Professor Mark Rosenthal and Professor Kate Drummond and others within the Brain Cancer Centre, we've developed this unique model, the Brainpop. And what this involves is a very broad, inclusive platform that involves patients, kids with brain cancer, adults with primary brain cancers, as well as patients with brain metastases from another cancer, and is developing a platform for advanced diagnostics that includes genetic testing of cancers as well as advanced imaging techniques, so that ultimately we can better predict predict treatments for patients. And that might include immunotherapy developed in the Jenkins lab, radiosurgery or gamma knife treatment with a new machine put in at Peter Mac recently, or precision medicine, trying to really come to that holy grail of personalised oncology for patients with brain cancer. By doing a second operation on treatment, we can then really be able to tell patients whether or not the treatment they're receiving is benefiting them, and then continuing on that therapy, or if not, providing opportunity to enrol in other treatments and to stop futile therapy earlier. This whole process can continue at progression, so we can then collect more samples from patients, and that process individualises and learns over time as well. So we can create a truly personalised approach for each patient. Now, for those of you with a lived experience of brain cancer, the idea of multiple surgeries, can be a little bit confronting. And what's important about the platform is that it's very inclusive. And whilst not everyone will choose to be a part of a perioperative program of work or may be suitable for it, the platform will be available for all people at different stages of their diagnosis. And that we hope, if we can build this right here in Parkville, that we can expand this model beyond to many sites in Australia and collaborate with colleagues internationally. And so to deliver on this, our team have long-standing collaborations here within the Melbourne Biomedical Precinct in Parkville and also beyond. And we're really actively engaging with new collaborations to meet the challenges. But more broadly, I think in Brain Cancer Awareness Month, it's very important for us to acknowledge that we're part of a bigger Australian brain cancer community that exists beyond this precinct, and that there's dedicated and passionate scientists, clinicians, consumers and patient advocates, and we're looking to develop lasting relationships. And change will only happen through embracing collaboration and partnership. So with that, I'd like to leave an acknowledgement slide and to acknowledge our fantastic team. We've just started in October, but we're really building a great network of young scientists who we hope will come together on us with this journey. Some excellent collaborators, some of them in the room here. A really dedicated consumer team who's informing our research program, and with significant thanks to Sam, to Doug, to Carrie and others in the leadership team. Thank you.
Thank you. Thank you so much to that group. Jim, do you mind to stay 1 second? Just in terms of, and Saskia and Sarah, I'll have to relay the question because you don't have a microphone, but the question that's just come up is, how did you three guys get together? How does a German fundamental biologist, a West Australian medical oncologist, how do you find out about this idea, get excited about it enough to say, actually, this could be my life's work? Because really that's what we want to inspire other people to do. So I don't know who wants to take that one.
Sarah, or do you want me to take it? Yeah.
Sarah's going to step up. So it is an interesting one because it's part of critical mass, right? How do we inspire people to come? So,
Yeah, no, I really love this question because I was finishing up my PhD in the Visveda lab working on breast cancer, and Jim sat next to me a few months before I left for my postdoc, and he was just starting his PhD on breast cancer as well. So we've known each other in the research space for some time. And I've known Saskia really well since I came back to do my second postdoc at WeHi. So we've had a really strong network for quite some time. We've known what each other are doing and what we're interested in. And and Jim approached us and told us that, look, it's time to switch focus. Brain cancer is really important, and together we can probably do something pretty amazing. And so it's what we're trying to do.
No pressure. Yeah. Thank you. And I think it's an important question because all it took was Jim's one thought to say, it's time to switch focus. And I think that's really important because it doesn't take a lot to change trajectory here. It does take momentum, and we've got some momentum, and we'll hear from other people through the afternoon, but you'd be surprised how little it takes to build the momentum to create a pathway towards our vision. And incredibly excited. So thank you so much, Jim, Saskia and Sarah, for your incredible work initially, and good luck in your work over the next 12 months, knowing it's been a massive week and few months for you guys. So thanks again. Thank you to those guys.